Hello. Now we will discuss about electron transport chain of aerobic respiration. Before going to this topic, we will see what steps are involving in the aerobic respiration. Aerobic respiration is completed in 4 steps. First one is glycolysis, which takes place in the cytoplasm. Second is pyruvic acid oxidative decarboxylation. Third is Krebs cycle. Fourth one is terminal oxidation. It includes again two steps: electron transport chain and oxidative phosphorylation. In aerobic respiration, terminal oxidation occurs at the end of catabolic process. It involves the passage of both electrons and protons of reduced coenzymes to oxygen. In this process, water is produced. Electron transport chain takes place on inner mitochondrial membrane. Inner mitochondrial membrane contains groups of electron and proton transporting enzymes. ETC or ETS is a series of coenzymes and cytochromes which take part in the transport of electrons. Inner mitochondrial membrane contains 5 enzyme complexes. Complex 1, 2, 4 means 1, 2, 3, 4 are involved in electron transport. The last fifth complex is connected with the ATP synthesis. Here the first enzyme complex is NADH dehydrogenase or NADH reductase. NADH ubiquinone reductase. It has two prosthetic groups. One is FMN, flavomononucleotide, and iron sulfur protein. Both electrons and protons pass from the NADH to FMN. Then electrons move to the iron sulfur complex and from there to ubiquinone. So here you can see the red color one is first enzyme complex that has two prosthetic groups. Electrons are moving from NADH2 to FMN, then to FES, from FES to ubiquinone. So the enzyme complex is exposed to outside and inside. Outside means inter membrane space, and inside means mitochondrial membrane. And the second complex, the yellow colored one, is second complex, succinate ubiquinone oxidoreductase. This enzyme complex has FAD, flavoadenine dinucleotide, as prosthetic group, and iron sulfur protein also. FADH2 gives its electrons and protons to ubiquinone through iron sulfur complex. So this second complex is transferring the electrons and protons from FADH2 to ubiquinone. Next is blue color one is Cytochrome C reductase. It has three components: cytochrome B, cytochrome C1, and iron sulfur protein. It reduces the cytochrome C. Cytochrome C is a small protein, so it reduces cytochrome C by transferring electrons via cytochrome 1, cytochrome C1. Here cytochrome C is named as cytochrome C reductase is named as it is transferring its electrons to cytochrome C, or it is reducing the cytochrome C. That's why the third enzyme complex is named as cytochrome C reductase. Cytochrome C is a mobile electron carrier. It transfers electrons between complex 3 and complex 4. So cytochrome C is reduced by the third complex, and cytochrome C is oxidized by the fourth complex. Next, the fourth enzyme complex is cytochrome C oxidase. It contains cytochrome A, cytochrome A3, and two copper centers. It transports the electrons from cytochrome C, reduced cytochrome C to molecular oxygen. Oxygen is a here ultimate electron acceptor. It becomes reactive and combines with protons to form metabolic water. Here, the fourth complex is donating the electrons and they are received by the molecular oxygen, and with the help of two protons, the molecular oxygen is producing the water, that is metabolic water. And energy released during the electron transport is available to specific protein complexes. Here you can see the first enzyme complex is transporting the protons, and third enzyme complex and fourth enzyme complex also. So these three complexes are involving in the proton translocation. So energy released during the electron transport is available to specific protein complexes. First one, third and fourth complexes. They can pump protons from matrix to intermembrane space, but not second complex. The complex 2 is it cannot pump the protons from matrix to intermembrane space. Here proton pumping creates a proton gradient. So the first, third and fourth enzyme complexes are transporting or pumping the protons to intermembrane space from mitochondrial matrix. So the proton gradient is established. The difference in the proton concentration creates an electric potential across the membrane. So this electrochemical potential gradient is called as proton motive force. This proton motive force is very important for the ATP synthesis. The protons which are pumped from mitochondrial matrix to intermembrane space, these protons again they try to move inside or try to enter the mitochondrial matrix. So they can enter through fifth enzyme complex only. While they are moving through fifth enzyme complex, here ATPs are synthesized. So the proton motive force is very important for the production of ATP. So that is about electron transport and proton translocation, and we will see the oxidative phosphorylation or ATP synthesis in the next video.